Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled How Cell-free mRNA Templates Support Faster Research with High Quality and Quick Turnaround. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank GenScript, who developed the content for this presentation. GenScript is the leading contract research organization in the world, providing gene, peptide, protein, CRISPR, and antibody. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make people and nature healthier through biotechnology by scripting possibilities. Now it's my pleasure to introduce our speaker for today's event, and he is Andrew Tsao. He is the Global Product Manager at GenScript, covering the molecular biology portfolio. His focus includes plasma solutions for key applications such as mRNA, AAV, LVV, as well as the development of innovative technologies that support cutting-edge research in gene and cell therapy, as well as synthetic biology. Now, before I pass the mic over to Andrew, we do have our first poll question that I'm going to share with you now. I'm going to launch it, and you should see it take over your screen, and there it is. Now, please go ahead and participate in this poll question as it's done in real time. So I'm going to read off the question to you. What is your biggest challenge when working with mRNA templates? So you have a couple of options below to select from. So please go ahead right now and make your selection. Is it maintaining poly A integrity during cloning and plasma prep? Or is it achieving high quality mRNA templates? Or is it long turnaround time with current supplier? And then the final option that you can select is high cost for current mRNA template supplier. So those are your options that you can choose from. So please go ahead now and make your selection. It looks like we got about almost half of our audience participating in this. So thank you so much for that. And for the rest of you, if you could please go ahead now and make your selection, I'll keep it open for another 10 seconds. And then I'll close the polls, share the results, and then we'll get right into this great discussion and presentation with Andrew. Okay, that looks like it's the majority of our audience. So thank you very much for that, everyone. I'm now going to close the polls, and let's take a look at the results. Okay, so here are the results. For our first poll question, here are the answers, or the selections that we have here. 22% of our audience said maintaining poly-A integrity during cloning and plasma prep, while 56% of our audience chose achieving high-quality mRNA templates, then 11% for long turnaround time and current supplier, and then another 11% for high cost for current mRNA template suppliers. So thank you very much, everyone, for participating in that poll question. Now, with that out of the way, it is now my pleasure to pass the mic and the controls over to Andrew. So Andrew, when you're ready, you may begin.
Hello, everyone, and thank you for joining today's webinar on how cell-free mRNA templates support faster research with high quality and quicker turnaround time. My name is Andrew, and I'm the product manager here at GenScript. I'm very excited to be here and very excited to spend the next hour with you. So our focus today is one of the most important foundations of mRNA research, the high-quality templates. Specifically, we'll look at how mRNA templates are constructed, the considerations of designing a mRNA template, and lastly, our newest cell-free template technology that's reshaping discovery timelines. Before we dive in, I'd encourage you to make sure to use the chat box below. Drop in any questions you have or comments as they come in mind. And I'll make sure towards the end of the presentation to address as many as I can. Here is a quick overview of what we'll cover today. Beginning from a brief introduction of GenScript, then we'll cover the current mRNA therapeutics landscape, the history of mRNA therapeutics, where it begins, and dive into the strategies and considerations when constructing an IVT template, designing a vector, the challenges that researchers face, and how and why quality matters for IVT success. Lastly, I'll introduce to you GenScript's innovative mRNA platform and how GenScript can accelerate your research. Okay, let's get started. So first, we'll start by a brief introduction of GenScript. So here's a quick overview, journey, and milestone of what we have accomplished over the past 2 decades. GenScript was founded in New Jersey in 2002 as one of the pioneers in gene synthesis. Over time, we've expanded into different new areas. In 2013, we've launched BestSign to focus on industrial synthetic biology. The following year, in 2014, we founded Legend Biotech, marking our entry into the cell therapy space. In 2015, GenScript was listed on the Hong Kong Stock Exchange. Then in 2017, Legend Biotech entered a strategic collaboration with Janssen on a new CAR T cell therapy. And in 2018, we established ProBio, our biologics CDMO partner. Now, fast forward to 2020, Legend Biotech was public, listed on NASDAQ. And in 2022, our groundbreaking CARVYKT Legend Biotech's collaboration CAR T cell therapy was granted approval to begin treatment of multiple myeloma. As you can see, GenScript really has steadily grown into a global group spanning over discovery, development, and also clinical solutions and therapeutics. Today, I'll be here representing GenScript Life Science Group, where we really provide a comprehensive range of custom services and products to our customers. We provide services from molecular biology, gene synthesis, plasmid preparation, to protein expression, to gene editing tools, mRNA, oligo, peptide, viral production, cell line engineering, antibody production, you name it. We really offer a one-stop solution to provide reliable research tools to our customers. So really at GenScript, we're not just supporting science, we are really continuing to support our customer to script new possibilities for the future. Over the past 20 years, we really built a truly global production network designed to really serve our customers closer and faster than ever before. We have production sites in United States, from our headquarters in New Jersey to our Seattle Innovation Center to Singapore and China. Also have regional offices worldwide, really ensure the supply chain resiliency, faster shipping to our customers. Okay, now let's dive into the main part of today's presentation, mRNA. To begin with, we'll start by diving into the mRNA therapeutics landscape, really exploring where the field is at today and where we came from and where it's heading in the future. So a brief history of mRNA. mRNA has really come a long way in just a few decades. The story really began in 1961 when Brenner and his colleagues described mRNA as an unstable intermediate that carries genetic information from DNA to ribosomes. This is the first time that mRNA was discovered, and it's also the fundamental discovery that laid the foundation for everything that has followed today. And then 7 years later, in 1968, a group of scientists demonstrated the first in vitro translation of mRNA, showing that mRNA could indeed be direct protein synthesis outside of the cell. This is also a really critical step in proving its potential as a tool for biotechnology. Approximately 20 years later, another major milestone was reached with the first in vitro transcription of mRNA, first using the sp6 polymerase. This breakthrough really meant that researchers could now generate synthetic mRNA in the lab, really then opening the doors to experimental applications. Fast forward to 1992, mRNA was first tested as a therapeutics. In a study, by Scripps, injection of vasopressin mRNA transiently reversed diabetes in animal models. This first early experiment showed that mRNA could be not only just a research tool, but it could also potentially use as a therapeutics to treat diseases. The true moment really came in 2020, where we all are really well known, is where the Pfizer BioNTech COVID-19 vaccine was approved. It was the first mRNA-based drug to receive approval. Let's now look in the future. In the future, we have really seen that the applications of mRNA are rapidly expanding. Expanding beyond vaccines, we are seeing innovation in cancer immunotherapy, personalized vaccine, in vivo CAR T, and other treatments to treat patients. The therapeutics really of mRNA and its potential has never been more exciting. Okay, now we've seen the history. Let's talk about what mRNA actually is and why it is so powerful for therapeutics. So mRNA, also known as messenger RNA, is essentially a genetic blueprint that carries instructions from the DNA and tells the cell which proteins to make. Structurally, as you can see in this diagram below, it consists of several key parts. The 5' cap that protects the molecule and helps with translation, the coding sequence that contains the actual protein, and then the regulatory regions such as the 5' UTR and the 3' UTR. And finally, the poly A tail at the 3' end, really to stabilize the mRNA and help it to function efficiently. So what makes mRNA therapeutics so exciting? There are three major features. First, it is programmable and scalable. Really, by simply changing the sequence of your DNA template, we can encode virtually any protein possible. This makes it incredibly fast to go from an idea to a therapeutic candidate, which is also what we've seen for the COVID-19 vaccine, much shorter than traditional drug development timelines. Secondly, mRNA is non-integrating. Unlike gene therapies that permanently alter the DNA, mRNA provides a more transient expression without altering the genome, which enhances the safety of the therapeutics. Lastly, it's really versatile. mRNA can be used, like we mentioned, in a wide range of applications, from vaccine to protein replacement, to therapeutic antibodies, and even to gene and cell therapies that we have seen today. Really, in short, mRNA is like a universal software program for biology, really flexible, fast, and safe. Okay, now let's dive into a bit of across the different therapeutic areas that mRNA have been used as a tool. At the center, you'll see the broad applications of mRNA drugs, ranging from vaccines to protein replacements, cell therapies, genome editing, and even therapeutic antibody production. Let's now walk through each one after the other. First, for therapeutic antibody production, instead of manufacturing antibody in a bioreactor, using mRNA, we can deliver and encode the actual antibody sequence directly into the body, using the patient's own cells to produce the antibody internally. This approach is much faster and cheaper than conventional antibody production. A really good example would be Moderna's mRNA 1944 candidate, which is currently in clinical trial, which encodes an antibody expressing the Chikungunya virus. Next, mRNA can also be used to do genome editing. Here, mRNA would be used to deliver genome editing proteins such as CRISPR-Cas nucleases in vivoli. The benefit is that we avoid DNA integration risks since the editing proteins are only expressed transiently. A clinical example would be Intellia Therapeutics drug for in vivo CRISPR therapy, delivering through mRNA plus a guide RNA in LNPs. Next, we have the protein replacement. Another really major application with mRNA is using mRNA to replace missing or defective proteins in genetic diseases, really by delivering mRNA with the actual functional protein to patient cells, which patients can produce themselves. An example would be Moderna's mRNA 3927 candidate for propionic acidemia, a rare metabolic disorder using protein replacement mRNA as a tool. Next, vaccines, probably the most well-known application. How it works is mRNA encodes the antigen that mimics parts of the pathogen, really training the immune system to recognize and to fight the actual real infection. These benefits are fast design and scalable for new variants, which we have seen and proven with our COVID-19 vaccines. Finally and lastly, cell therapies are really an application that recently have gained a lot of interest. mRNA can be used to reprogram immune cells such as T cells or NK cells for cancer therapies. This advantage is now safer and more flexible compared to DNA-based method. A really prominent and recent example is Capstan Therapeutics, a new CAR T therapy against B-cell autoimmune disease. Really, together here, what we're trying to demonstrate is to show how broad the therapeutic potential of mRNA really is. It really is not just about vaccines, but a whole new toolkit for medicine and therapeutics. Okay, now we have seen the wide range of applications for mRNA therapeutics. Let's dive into and look more closely at one of the most fundamental part of this field, the IVT template. Where really is the mRNA where it's made. In the next sections and slides, we'll walk through the different ways of constructing an IVT template, the essential elements of IVT template design, key considerations, and highlight the common challenges that researchers face today. And really, lastly, explaining why high-quality template matters for IVT success. So really, it's simple as it looks. To make mRNA in vitro, we start with a linearized DNA template that contains all elements needed for transcription. And with the help of a RNA polymerase, NTP mix buffer, and the appropriate buffer, the template is then transcribed into RNA. At the same time or after transcription, we can introduce modifications such as capping to improve stability and really to mimic the natural mRNA. However, while the workflow really looks really straightforward as it is here, the quality of the DNA template is ultimately determines the efficiency and the reliability of the IVT process. Really poorly designed and unstable templates can lead to low yield, truncated mRNA product, and inconsistency, which is why we're demonstrating that template design and quality is so critical. There are different ways researchers approached when generating IVT mRNA template. Here we're demonstrating three main approaches today. The main difference lies on the way you introduce the poly-A tail. So first is the post-transcriptional poly-A addition. Here, what you really do is you make the RNA first, and then use poly-A polymerase to add the poly-A tail afterward. The main advantage here is speed and flexibility, since you don't have to clone long stretches of poly-A into your DNA. However, there are major drawbacks with this method. The poly-A tail length is hard to control and inconsistent. There are high batch-to-batch variability. And since we're using the polymerase to add poly-A, the cost can add up really quickly, which makes this process not as popular as the two other methods. Secondly is the plasmid template that actually encode the poly-A tail in the template. This is the mostly widely adopted approach, especially adopted in clinical and therapeutic use. Here, the poly-A tail is encoded directly in the plasmid. So every transcript would have a defined and consistent length. There is high reproducibility and high batch-to-batch consistency, and of course, well adapted in clinical applications. There are some cons and downside of this method. Cloning and maintaining long poly-A stretches is very challenging in E. coli. Risks of recombination and really unstable when propagating in E. coli. Lastly, we have the PCR-based poly addition method. So here, instead of cloning or adding tails enzymatically, we use PCR primers, which encode poly-A tail to amplify the actual template. This avoids plasmid instability issues and really is preferred and work well with rapid screening and small-scale studies. However, there are also some drawbacks of this method. Firstly, it is not scalable for larger productions. And also, we've seen heterogeneity in tail length with the PCR method. So in conclusion, really while all three of the workflows look similar, the actual method you choose for poly tailing really has a major impact of your downstream success. This is really important that you want to optimize your workflow and determine the best template design upfront. It's so important. Okay, now we have compared the approaches. Let's look at how actually a IVT template is designed for success. Think of it as moving from left to right of the template, from the 5' end to the 3' end. So first, you will start with your promoter. The promoter, this is where your transcription begins. T7 promoter is by far the most commonly used for IVT applications. While alternatives such as SP6 and T3 promoters are used, but less common, and typically only used in specialized applications. Next on the line, you have your 5' UTRs, the untranslated regions. What 5' UTR does is it enhances ribosome binding and increases translation efficiency. Here, the most widely used UTRs are wild type HBA and HBBs. Which have been proven with its ability and stability. However, it's worth noting that most companies, therapeutic companies, do have their own synthetic UTR sequences that they have engineered to have their tailored benefit. Also, something that we have seen recently is that researchers have started to adapt tissue-specific UTRs when targeting specialized tissues and organs for specialized expression. Next, we have the coding sequence, which is pretty straightforward. It would be the sequence that actually encodes for your protein of interest. After your coding sequence, you have your 3' UTRs. What 3' UTR does is it regulates transcript stability, localization, and affects translation efficiency. Again, the HBA, wild-type HBA and HBB is widely used for its proven ability. And also noteworthy that companies do have their own synthetic UTR sequences that they prefer. Lastly, in your template is your poly-A tail, which functions to protect, enhance the stability and translation. For most therapeutic applications, the standard range is around 100 to 120 poly-A's. Shorter tails, around 80 A's, are sometimes used in screening, where the scalability matters more than the durability. It's worth noting that poly tail is one of the most important parts and also the most challenging part of the template, since, like mentioned, poly tail are known to be unstable in E. coli during cloning and plasmid preparation. That being said, it's recommended, and most labs and researchers rely on specialized strains and specialized production processes, or even segmented poly designs to preserve the poly fidelity during plasmid preparation. After learning about the key elements of your IVT template, I want to really dive into the poly tail. Since we mentioned it really plays a critical and central role in the IVT success, and also with the given challenges mentioned. When looking at poly tail, really there are three key main considerations when designing and optimizing your tail sequence. First, the fidelity and the stability of your poly tract is absolutely critical. If you lose length or introduce errors during plasmid prep or cloning, you'll see a direct impact on your IVT efficiency, and ultimately that will affect your mRNA performance and mRNA expression. Secondly, choosing the right tail length and design really makes a big difference. Like mentioned, most therapeutic RNA is typically adopt 100 to 120 poly-A's, which is long enough to support strong expression and stability. But for some screening applications, shorter poly tail may be used to increase the stability, and also segmented poly designs can also help with the stability and reduce risk of the recombination in poly tail, while also maintaining the expression. Lastly and thirdly, really even the best design can fail if you don't use the right tools. So it is always recommended that we use a specialized stable bacterial strain and for your production process, really to maintain and to preserve your full poly tail integrity. With all of that being said, you can really see on the internal study that we have done at GenScript we have different poly tail designed, and we've run GFP intensity at 48 hours to look at how the poly tract affect mRNA expression. As you can see, the 120 and 100 A's tract have the highest expression, followed by segmented versions, and as the poly tail shortens, you see decreasing mRNA expression. So really the key takeaway is mRNA design is not just simply about adding editings into your template. It's really about a balance of maintaining integrity, choosing the right length for application, and also the right tools to propagate your mRNA template to really safeguard your downstream studies and work. So other than poly integrity, there are actually much more factors that would affect your IVT performance. Now let's zoom in or zoom out to the full picture of your template quality controls. Really, every step of IVT success ties back to your IVT, the quality of your IVT template. So there are different QC metrics that we often consider when we look at your template quality. First, and really most importantly, is your sequence fidelity of your insert. Really, any mutation in your sequence fidelity would directly impact your protein translation accuracy. Really, a single base mutation can really alter codons or regulatory elements that will affect successful IVT and downstream work. So really, sequencing verification is really important to ensure successful experiment. Next, poly tail integrity. We have mentioned this a lot in previous slides. Really, poly tail is really important for mRNA stability and translation efficiency. When you see truncated poly tail, you see directly affecting the mRNA expression. Next, endotoxin level. While endotoxin from our data does not directly affect IVT reactions, the really critical here is that the endotoxin can carry over into the final mRNA product, which is really unacceptable for downstream applications, especially for therapeutics or animal studies. So it is recommended to always use endotoxin-free plasmid kits and optimize purification workflows to ensure the endotoxin removal when preparing IVT templates. Next, linearization purity. Really, what linearization purity means is how efficiently did you digest or cut your plasmid DNA. You really only want your linearized plasmid to ensure successful IVT experiments. Incomplete digestions or circular plasmid residues can lead to heterogeneous transcripts, which really directly lowering your mRNA purity and yield. We always recommend to use high-fidelity extraction enzymes, optimize your digestion conditions, and carefully QC your templates either by agarose gel or capillary electrophoresis to ensure complete digestion before IVT. Next, host DNA contamination. Since most researchers often use E. coli for cloning and plasmid DNA, plasmid preparation, genomic DNA, and other host-related contaminants from the bacteria host may carry over and compete with your template and really affecting and lowering your IVT efficiency. That being said, always carefully purify your templates before IVT. Lastly, protein contamination. Really, protein contamination can come from many different perspectives. It can come from carryover from your host, come from accumulated enzymes from your intermediate processes, which all can affect IVT performance. These residual enzymes and proteins can degrade the transcript itself and also interfere with IVT. We always recommend protease digestion, really stringent washes and purifications before IVT reaction. And if available, always run ELISA or protein assays or even nanodrop to confirm that your DNA is pure without any protein contamination. Really, the big picture that we're showing here is that high-quality IVT really starts from high-quality DNA, and all different quality control metrics matters for your IVT experiment. Here, we are moving from what we have talked about, the theory, into actual data. At GenScript, we have run controlled experiments to quantify how different template impurities and quality metrics might affect or impact IVT performance. And the results are really as expected. Now let's first look at linearization purity. So when digestions are incomplete, IVT drops and your mRNA purity really suffers. You can see at 50% digestion, mRNA purity was just at 11%. But once we near complete digestion, you see the mRNA purity rose to above 80%. This really shows how critical clean linearization is for high-quality mRNA transcripts. Secondly, host cell contamination, how it affects IVT performance. As you can see, with 25% of host cell DNA contamination, you can see that the mRNA purity drops below 80%. When reducing your host cell DNA contamination to below or at 1% or even 5%, you see a really good pure DNA purity above 86%. This is a clear sign that host DNA would affect your IVT performance during transcription. Lastly, protein contamination and how it affects your IVT performance in your mRNA. At 15% protein contamination, purity is only at 80%. And when you lower the protein levels to below 5%, 2%, or below 1%, you see the mRNA purity rises to around 85%. So the overall takeaway here is really to show that actually your template quality affects your IVT success and really reflects in your mRNA quality. So really, when you're considering your template quality, carefully perform QCs and ensure and make sure that you have your clean starting material for your IVT process to make sure that you have your final mRNA product consistent, pure, and really usable for your downstream applications. As we wrap up, let's leave some key messages. First, template quality is really the foundation of IVT success, really from sequence fidelity to impurity, contamination to poly A, tail integrity. Everything directly impacts your RNA yield and stability and performance. Secondly, the design of your template matters as much as your preparation and quality. It's about the right mRNA, really from your promoter to your optimized UTR to ORFs and the poly A tail. And lastly, really ensure that you perform comprehensive QC to safeguard your mRNAs. Rigorously check for sequence accuracy, poly A tail integrity, and also contaminants to ensure that you have clean, high-quality templates for your IVT mRNA production, whether it's for your discovery, research, or downstream animal or preclinical studies. All right, so that's the end of the template introduction. And really here we want to move into the main part of the presentation. How does it all come together in practice? And really introducing at GenScript our end-to-end mRNA solutions. And really showing how at GenScript we are not just providing a service, we are partnering and we're dedicated to enable our customer success at every stage of mRNA development. Okay, so let me take you through our offerings. Really, for over 5 years, GenScript have been the largest gene provider in the US, really trusted by over 200,000 customers worldwide, and that numbers continually to grow. Really, the trust is earned by us consistently delivering results with an on-time delivery rate of over 95%, and with our unmatched production capacity globally in New Jersey, United States, Singapore, and China. And when speed is critical, we lead the market in providing fastest service possible. Our clonal genes is shipped starting from just 72 hours, and we have delivered over 4 million genes and counting. Really, GenScript is uniquely positioned to help you accelerate discovery and scripting the possibilities of your research. Okay, now let's dive into mRNA solutions and what we offer in terms of mRNA. What makes GenScript unique is that we can take you from gene design all the way to mRNA synthesis to LNP particles, all under one roof at GenScript. So it starts with codon optimization design. We offer with our GenSmart codon optimization a proprietary algorithm that screens over 200 factors to ensure that you have your highest protein expression possible. At GenScript, we also have PhD level technical scientists to support your project, your design of your mRNA. From there, we move into gene synthesis. GenScript, we are best and most well-known in the industry in synthesizing complex sequence. We do sequences up to 200 kilobases and turnaround starting at 3 business days, all with 100% sequence accurate genes. Next come cloning and plasmid preparation. For mRNA, we offer ready-to-use proprietary mRNA vector backbones and plasmid preparation and linearization, different grade options supporting your basic research all the way to your high-quality needs for in vivo animal studies. And lastly, we also have our newest exciting offering of our cell-free mRNA template service, which we'll dive into shortly. From IVT templates, we also offer mRNA synthesis, different types of RNA, linear mRNA, self-amplifying mRNA, and also circular mRNA, from RUO to preclinical GMP-like grade. And finally, we complete your journey with LNP lipid nanoparticle service, providing we have different targeted LNP formulations for your needs to target your tissue of interest. But for today, we will only focus on GenScript solutions for IVT mRNA template generations. Really, when talking about mRNA templates, GenScript has built a complete ecosystem to deliver innovative solutions to deliver highest quality mRNA templates to our customers. Now let's look at what really set us apart. First, our poly guarantee service is an add-on service to our plasmid projects. We have developed a specialized production process with our proprietary GenStable poly strain to ensure the exact actual length and high purity of your mRNA poly template. Next, we offer ready-to-use mRNA applied vector, specially engineered with pre-built poly sequences. These are engineered to have superior ability to retain poly integrity and also with increased plasmid yield. Finally, we have our newest cell-free mRNA template service. With this really breaking innovative cell-free technology, we deliver IVT-ready templates in record speed. Really developing this with our customers, mRNA researchers in mind, delivering fastest mRNA template for customers with tight deadlines. Together, all of this is what makes GenScript the clear leader in delivering mRNA template solutions. Now let's dive into one after the other. Starting from introducing our mRNA applied vector, we have ready-to-use vectors where customers can directly select for their cloning projects. At GenScript, these come in different formats, differing from poly tracks as well as cleavage site. We have different poly tracks, 100As, 120As, and also segmented versions. We have engineered our backbones to really surpass conventional limitations that researchers are facing today. There's three main advantages and benefits of our vector. First, they have superior poly stability. Secondly, they have superior plasmid production. And lastly, high purity in producing monomeric supercoiled plasmids. Starting from poly stability on your right, here we really showed the ability of our vector in maintaining poly integrity. We compared our mRNA vector to Pac57 vector versus our mRNA applied vector. Both we constructed with different insert containing poly tails of 100As and 120As with a GFP insert and other different inserts. And these were cultured in our GenStable poly strain as well as our NEB stable strain over 8 different generations. And Sanger sequencing of the poly tail was conducted at each generation to evaluate the integrity of the poly sequences. Here, we're showing the percentage of each generation with the complete intact poly tail length. As you can see, our mRNA vector with GenScript GenStable strain over 8 generations, we did not see any poly tail loss. And also, while using either one of our solutions, we also see a better poly stability over time, comparing to conventional solutions using Pac57 and an EB stable strain. Next, if you look at your bottom left-hand graph over here, here we demonstrated that our vector have demonstrated 100% increase in plasma yield comparing to the conventional Pac57 vector. Here again, genes with different various inserts are cloned into our backbone and the Pac57 backbone containing 100 and 120A poly tails. Following overnight propagation, maxi prep, plasma yield were extracted and evaluated. You can see here that our GenScript mRNA vector had over 100% increase in plasma yield versus the conventional Pac57 vector. Lastly, on the bottom right-hand side, here we demonstrated that our vector's ability to get clean monomeric supercoiled DNA, which is actually crucial to ensure that we have consistency across batch-to-batch mRNA template production. This is actually very critical. A lot of our customers have mentioned that very critical for regulatory submission and approvals. You can see we also have different inserts. Here we also include the toxic and unstable inserts and clone them into our mRNA vectors versus the PAC57 conventional vectors. And here we can see that our vectors has consistently show over 30% more monomeric supercoil percentage when compared to conventional PAC57 vector. Here you can also see on the gel electrophoresis graph that our vector you show a clear single band versus PAC57. You have different isoforms and of the supercoil versus our vector, really a clear monomeric isoform of DNA supercoil. Really, to summarize, our mRNA vector isn't just another cloning vector. It is really specially engineered with our customers in mind to deliver reliable and high-performance mRNA templates that save time and money. When our customers use this vector and bring this to downstream applications to preclinical and clinical studies to save them time and money, also improve reproducibility as well. Next. I'm sure if anything you have learned so far today, probably the biggest getaway is the pain point in mRNA template preparation and maintaining poly-A tail integrity. This is exactly why at GenScript we have developed a poly guarantee service, which is an add-on service available for your plasmid projects. When added on, your entire project will be handled by a dedicated and experienced team using the protection of the gen-stable poly-A strain that we just showed in the previous slide to ensure superior poly-A integrity. And when bundled as part of our quality control and release standard, we will perform Sanger sequencing, poly-A sequencing of your entire poly-A tract to ensure that you have your complete exact length of your poly-A sequence. Here, really, to deliver the highest purity verified poly-A templates to give our customers the full confidence in their downstream workflow. Lastly, and most importantly, today's main topic, our newest and groundbreaking service, the Cell-free mRNA template service. We have developed this service really based on many feedbacks of our customers, really with the need for faster template generation for their stringent timelines, especially in discovery screening phases. We are really proud to say that we have delivered. We have delivered with our Cell-free service. With our Cell-free service in overview, we can deliver IVT-ready templates in as fast as 3 business days, and from gene synthesis in as fast as 6 business days, with the same high quality that we have always delivered. The process also starts with gene synthesis and cloning. Instead of the time-consuming E. coli-based amplification, the templates are here then amplified enzymatically using a specially GenScript-developed high-fidelity 5C29 enzyme, which completely we will bypass bacterial system, eliminating any wholesale bacterial contaminants. Following amplification, from there we will perform enzymatic linearization to ensure complete linearization of your template to get individual copies of your DNA template. Following linearization, we will perform a streamline purification to remove any residual proteins and impurities that are from the intermediate processes. And then every single template will undergo quality control to ensure that your insert sequence is 100% of your design, your poly integrity is exactly what you desired, and also fully linearization confirmation to ensure the highest quality and highest input of your IVT DNA. The result here, what our customers are really getting is a IVT-ready DNA, which they can directly use in their IVT experiments without any additional purification steps required. And again, all of this can be performed in just 3 business days with our Cell-free process, really designed for these early discovery screening mRNA customers who demand ultra-fast turnaround time. Since the entire process is cell-free, We also see really high pure DNA templates. Here we are demonstrating and showing the quality of what these cell-free templates demonstrate. And this is data based on over 60 different templates with different inserts that we have generated internally for beta testing and also alpha testing and also beta testing from our customers. 100% of the templates demonstrate these metrics. Endotoxin below 0.05, EU per microgram, there's absolutely no RNAs detected, total protein below 0.5%, and host DNA below 1%. Here we also show that part of our default QC options, DNA template, 100% linearization, BAL analyzer of the DNA template purity, a single clear peak, and also folate tail verification via Sanger sequencing. You might also wonder if and how does the IVT mRNA quality compare to traditional templates? We have also done thorough analysis and study comparing the two methods. Here we are showing some representative results based off our finding, but the findings are really consistent across all of our tests. On the left, you can see that the IVT mRNA from the cell-free templates show clean, high purity bands on the gel. This is confirmed again in the middle, where a BAL analyzer demonstrate tight, reproducible purity profiles across all replicates, and also comparing to conventional plasmid templates. And most importantly, on the right, this then translates into strong and consistent protein expression. Here in the mCherry encoding templates assay, mRNA synthesized with mCherry inserts were translated and synthesized, which we show that deliver robust expression in both 24 and 48 hours. Also fully comparable with the control, which is the conventional traditional linearized template. Hey, I hope everyone have a brief and a thorough understanding of our cell-free service and is as excited as I am and ready to try our new service. Really feel free to reach out to us after the webinar if you have any questions and would like a pilot test of this service. Really lastly, as a conclusion, as we've seen today, GenScript platform really gives you everything you need to unlock your full mRNA research. First, we provide a comprehensive solution spanning from gene to IVT template to mRNA synthesis and finally LNP delivery. Really with GenScript, you have a true one-stop solution for all your needs. Secondly, we provide innovative technologies to ensure high-quality templates from our proprietary mRNA backbones, our Poly Guarantee service, and our newest cell-free technology to enable and ensure the highest quality in the most fast turnaround time manner in as fast as 3 business days. Thirdly and lastly, and very importantly, is expert support. We have PhD-level scientists to provide consultation on template and project design, as well as dedicated project managers to oversee all your project timelines and communication. Together, this means that you will get you will get your comprehensive support and need, more confidence, more reliability to focus on your research. All right, that concludes our webinar for today. Thank you so much for your time today. I've included my email here below. Feel free to reach out to me with any questions or if you'd like to learn more about our service. Now back to you, Sonia, for our Q&As.
Well, thank you very much for that very engaging and insightful presentation, Andrew. We're going to jump right into the last poll question right now, and then we'll get right into the Q&A. So remember, audience members, you still have the opportunity to send your questions through using that questions window. And here is our poll question. Please go right ahead and make your selection. Would cell-free mRNA templates be of interest to you? So please go ahead, everyone, and cast your vote. Is it yes, I have immediate order needs? Yes, but I don't currently need templates, or is it no, not relevant to my research, or no, I'm not interested at this time? So please go ahead. I'll leave it open for a few more seconds to get everyone involved in this poll question, and then we'll go right into the Q&A. Okay, perfect. I'm going to close the polls as we got to jump into the Q&A. Let me just share the results very quickly here with everyone. And look at this here, Andrew, 43% are saying yes, I have immediate order needs, while 57% are saying yes, but I don't currently need templates. So let's get right into the Q&A. Thank you very much, everyone, for participating with that. And now the Q&A will begin. So I will put on my webcam and invite Andrew to put on his webcam. And there we are. Hello, everyone. And let's see here what we have. Okay, just as a reminder to everyone, hi Andrew. As a reminder to everyone, if you are attending this via GoToWebinar, please use the questions windows. And for those of you watching on LinkedIn Live events, feel free to post your questions in the comments section. And I received a bunch of questions already, so I think we have time for maybe one or two. So let's jump to this question here, Andrew. This audience member is asking, can you share more about the performance of the mRNA applied vector in terms of IVT mRNA expression and overall mRNA quality? Any limitations researchers should be aware of in terms of use and whether commercialization would require a license? A great question. Your thoughts, Sir Andrew?
Yeah, great question. Yes, we actually have conducted a lot of internal studies and also feedbacks from our customers who really love our vectors. And really all of our internal studies and feedbacks from our customers showed and that not only our vectors can pertain the integrity of the poly-A, but it also has equal or even better mRNA quality produced from our vector. And the second part of the questions in terms of the usage for research use, there's no licensing required for any research use purposes. But however, if our customers wish to bring the vector into commercialization or to clinical trials, then it will require an official license with GenScript.
Okay, thank you so much, Andrew. I'm going to squeeze in one last question. Since you mentioned that your cell-free template service is positioned for early discovery screening customers, do you also provide late discovery or animal study grade material? And what yield options are currently available? Have you evaluated dsRNA content of your mRNA generated with the cell-free templates? Sorry, they had a lot of questions packed into that one.
Yeah, I'll try to answer this one after the other. But
Okay.
yeah, thank you so much for the question. And yes, for the current service that we are offering right now, we are only offering up to 500 micrograms of the IVT-ready templates, the linearized DNA, and at research grade quality. And if our audience are familiar with GenScript service, the quality is comparable to our tier 2 grade. But of course, at tier 2 grade, it's already of very high quality, but we do have customers' feedbacks during beta testing and also through the first couple of weeks of launching the service that they need the same fast turnaround time, but for their more downstream animal studies. That said, we are planning and working on a higher yield and higher quality service offering for these customer needs. And just the last part of the question, yes, the double-strand RNA content, that's something that our customers and researchers really care about. We have done testing using our current cell-free grade templates and look at the mRNA double-strand RNA and the mRNA generated, and they consistently show below 0.5% of double-strand RNA, which is low and also acceptable for these early discovery customers. But of course, we have the plan to upgrade the quality and of course the yield as well. Yeah, coming soon.
Okay. Well, thank you very much, Andrew, for trying to get those answers in. We truly appreciate it. We've come to the end of the Q&A portion of the webinar. And for those of you interested in hearing more from GenScript, you can register for their next webinar, High Titer Lentivirus Solutions in 5 Days, Break Production Barriers for a CGT scheduled for September 4th. I put the links in the chat box already, so you have it there. And if you have any further questions, please direct them to the email address on your screen. And just to let you know, we had a whole bunch of questions that more questions that came in, but unfortunately we're unable to answer them now. But be rest assured that the team at GenScript will follow up with you after this presentation. Now, thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive in the for this event, which you see in your chat box there. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, this, as I said before, this recorded version is in the chat box there. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event to gain access to viewing the recording and more. Now, please join us in thanking our speaker for his time here today. So thank you so much, Andrew, for your time. We truly appreciate it.
Yeah, thank you, everyone.
Okay, we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. I'm Sonia Hunt. Until we see each other again, please take care and bye for now. Bye, everyone. Bye, Andrew.
Thank you.